Good afternoon, everyone. Thank you for the kind introduction, and thank you for the opportunity to present here. It's been a very interesting conference, and I've learned a lot. And it's also a pleasure to present after some of the eminent speakers in this session, Prof. Toy, Prof. Rosen, and I think this is a great, hopefully my talk would be a great continuation of the themes that they've discussed. In particular, nanopore sequencing was a recurrent theme, and it's widely used nowadays to do whole genome sequencing of isolates. But also there was talk about nanopore sequencing for metagenomics. What I'll be talking about is to take the next step further, which is to take the nanopore sequencing of a metagenomic DNA sample and then tease apart whole genome sequences of individual bacteria from that. That can be used then for the transmission analysis as well as understanding antimicrobial resistance patterns. Okay, so I'd like to start with kind of a theme for what the lab does. The lab is interested in studying the role of the microbiome as a mediator, so the microbial communities in general, and how they sorry about that, how they influence the body's interaction with pathogens. And we look at various technologies to improve our studies of these microbial communities. So being at the Genome Institute, we see this from a genetic perspective, where we want to be able to read DNA, we want to reveal the function of DNA, and write DNA potentially as a therapeutic strategy. And given that we live in a microbial world, and the fact that DNA is sort of the ultimate barcode for microbes, and they have a vast untapped biodiversity, we think this is a very powerful approach to study microbial communities. But there are several challenges in existing microbiome studies and use of metagenomics for infectious diseases. And this is really the fact that we don't have whole genome information from a lot of the microbiome work that we do. And because we know that microbial communities interact with each other, the pathogens in a microbial community will interact with the commensals, we need a systems understanding of how microbial communities evolve. And finally, we need to really leverage the full power of metagenomics as a unified framework for rapid identification. Our current strategies have weaknesses in terms of the information they provide, the speed, and cost of analysis as well. So this is really a question about how we are evolving metagenomics as well as sort of microbiome studies from the classical marker gene-based studies, and others have referred to how these are give limited information, to the shotgun metagenomics, short-read shotgun metagenomics that has been common in recent years, but still leaves us with a bag of genes kind of information, to the third generation, these long-read based metagenomics, and I'll get into more details. So why do we really need this for the infectious disease studies that we are involved in? So this is a study that we are currently involved in various collaborators, where we're looking at how gut colonization by multi-drug resistant bacteria, so carbapenem resistant Enterobacteriaceae, can lead to creation of a reservoir that can facilitate onward transmission. So we are interested in understanding how the gut gets colonized, and how the community of bacteria in the gut might promote decolonization, so reduce the persistence of these bacteria in the gut. So some of our preliminary data that I'm showing you out here, on the y-axis is the abundance of various Enterobacteriaceae species, and the x-axis are the different patients at multiple time points serially over a year. And what you can see here, just based on the differences in colors, is that there's many different Enterobacteriaceae species typically in these subjects. The abundance of Enterobacteriaceae species is quite high, very unusual compared to a healthy gut microbiome. And there's quite rapid dynamics, so the color changes over time, and there are cases where a Klebsiella pneumoniae colonization is followed out by an E. coli colonization, again clonal or high dominance of these bacteria, but there's rapid shift in the community structure. What we also notice is that in addition to the fact that there's change in species composition, even within a single species, there are often multiple strains. So while E. coli is a commensal species, There can be the CRE E. coli that we observe is a distinct strain from what was there in the gut. In addition, even for Klebsiella pneumoniae, we often see multiple strains of Klebsiella pneumoniae in the gut. So we need strain-resolved metagenomics and be able to do whole genome assembly. What we really want to do is leverage long-read protocols. Now, there are multiple challenges in how we can do this. First is the optimizing of workflows, but also it's in terms of assembly. So I'll get into the assembly questions next. But here's a brief preview of nanopore sequencing. If you've not seen this before, the concept is very simple. There's a portable device that we can use for sequencing where DNA translocates through a biological pore, and the translocation inhibits the flow of ions, and that can be read out as an electrical signal that we decipher into sequence information. So the power of this technology is really the fact that it produces multi-kilobase pair reads, and this information is accompanied by also a higher error rate, as others have referred to. But this high error rate, while it's high compared to Illumina sequencing, is getting better over the years. So you can see how the sequencing quality has shifted and is now approaching, say, something like Q10 and Q20, which is quite useful for some of the work that we're planning to do. So what do we do with these long reads? And in terms of assembly, what we're trying to do is basically solve a giant puzzle. It's a one-dimensional puzzle. The picture here is trying to give you an idea what a two-dimensional puzzle would do in terms of piecing a genome. And this puzzle process begins with the idea that once you have sequencing, so you converted the DNA information to reads, the reads might carry signals of overlap, so regions which are shared between the reads. And this overlap information can be captured in a graph, what's called an assembly graph, where the reads are nodes and an edge connects two reads if they overlap. So the idea here is that if you can walk through this graph of reads where there's overlap between them, the walk gives you a sequence that could be the potential genome that is there in your sample. Of course, there are several challenges. You might not have enough coverage. You might not have covered a certain region of the genome, and so the walk might terminate abruptly. There might be issues where there are repeats. There are regions of the genome that are repeated across your reads, and so that might lead to ambiguity in terms of paths. You don't know which path to take in this graph. And finally, in metagenomics, you're not talking about one path, you're talking about multiple paths, because every bacterial genome will give you a distinct path, but there might be some shared regions between them. So metagenomic assembly is perhaps one of the most challenging assembly problems out there, and this is an area that's of active interest in the assembly and bioinformatics community. So we considered this challenge and we wondered whether we can do strain-resolved metagenomic assembly, where we can tease apart, say, multiple Klebsiella strains in the gut microbiome of an individual. And one of the innovations we propose is that this problem cannot be just thought of as a clustering problem. Classically, it's been thought of as a clustering problem where you use the coverage of contigs in your assembly and just cluster contigs which have similar coverage. So here, the nodes that are shown with similar color. But what we can actually see is that this is actually an image segmentation problem. So you actually have edges between the contigs which are given by the assembly graph information, and the color tells you the coverage of the contigs. So you want to segment the graph such that you have a bunch of contigs, a bunch of nodes which have edges between them, so they are assembly connected, but they also have similar coverage patterns. The other thing that we wanted to leverage is the fact that through the efforts of people around the world, we have sequenced isolates for many species out there. So we have very large reference catalogs, and these reference catalogs provide us prior information on how genome structure looks for various bacterial pathogens. So we synthesize some of this information into an assembly algorithm. I won't go into further bioinformatics details, but this method we call Opera MS, and we published this a few years ago. So what I'll try to share with you is the results from this approach and perhaps convince you that this is a useful way in which we can do study complex microbial communities and still do whole genome analysis. So we benchmark our method against many of the state-of-the-art methods. Many of them are based on Illumina sequencing, so that's what you can see on the x-axis here. On the y-axis is the improvement you can observe using this hybrid assembly approach. And the colors here show you the coverage of the particular genome that you're interested in. So the green and blue are higher coverage, and as the coverage drops to something below 10x, so very low sampling of the organism, the assembly performances obviously suffer. But what you can see here is that if you have sufficient coverage, our approach improves over just pure Illumina metagenomics by a factor of 10, so more than 1,000% improvement in many cases. But even when you have just the long reads and you have conventional long read assemblers, it still provides a 200% improvement over conventional assembly approaches. So having a targeted hybrid assembly algorithm provides a significant boost in assembly. Instead of getting small contigs, which are a few kb long, now you get contigs which are hundreds of kb long, enough that you can do something called binning and actually reconstruct whole genomes. And the other feature of this approach is that you can get strain-resolved assembly, and I'll highlight an application of this. So as I mentioned, we had a study where we were looking at gut microbiomes of individuals colonized by multi-drug resistant bacteria. In this case, this was an E. coli colonization, and we soon realized that there are actually 2 strains of E. coli, and the assembly actually also spit out 2 genomes out of this sample. So once you have this genomic information, what you can then do is try to track where the resistance determinants are. So in this case, the resistance determinants were in a plasmid. And so we had the plasmid genome and we had the strain genomes. Classically, it's assumed that you cannot relate the strain with the plasmid without some other information, without an isolate, or without using some sort of proximity ligation information. But in this case, what we showed is that if you have longitudinal data, and which is what we had in this case, and you had these strain-resolved genomes, in these 2 strains, the one on the left here and on the right, only one of them has a coverage of the genome and the plasmid that's highly correlated. So this high correlation indicates that the 2 genomic elements actually go together, and so that particular strain actually bears the plasmid of interest. So we can confirm this. Of course, we confirm this using an isolate that we had acquired for the same sample. And this confirms that the strain that we had reconstructed is accurate, very similar to the isolate that we observed. In addition, having metagenomic assembly allows you to discover biology that would have been very difficult and painful to reconstruct using isolates. So one example is understanding plasmid and phage diversity in various samples. So in this case, we were able to reconstruct closed circular genomes for about 8.9 megabases of plasmids and phages from 28 stool samples. And they cover a wide range of sizes shown on the left here in this figure. So all the way from a few kb to hundreds of kb. And many of these plasmids, as you can see on the figure on the right, had identity which was very low compared to what's in existing databases, and even the coverage of these sequences was low. So we were quite intrigued by this, and in fact it turned out that one of the largest sequences that we have assembled that we thought was a plasmid or a phage turns out to be actually a phage that has no homology to existing phages in the database, and it's what people call a jumbo phage because it's 263 kilobases long, and it's one of the first examples of human gut jumbo phage that is automatically assembled from metagenomic data. So now that switching gears to environmental microbiomes. Environmental microbiomes are particularly hard to study because of the low biomass of the samples that are typically studied. And so in a study where we were looking at environmental microbiomes across the hospital that I discussed yesterday, we had samples from more than 179 sites, 355 microbiomes. And we found that, in fact, if you try to culture them, you get a diversity of cultures. Many of these cultures are polymicrobial, and we wanted to study the genetic information in these cultures. So we decided to go ahead and sequence them using nanopore sequencing, and we found that this approach was able to capture a lot of the pathogen pathogen species that we are interested in, even though the abundance of these species can be quite low in the sample. And then using this hybrid assembly approach, we observed the assembly quality is very high. So we again get N50s in the megabase range. So the genome is in a few pieces that we can put together rather than the thousands of pieces that you can typically get with metagenomic assembly. And really the key result that emerged from the fact that we had very high quality genomes from a metagenomic sample was the fact that these genomes could then be put together into transmission or phylogenetic trees that give you some evidence of some information about relatedness and potential transmission pathways. And when you overlay the resistance information on these genomes and these phylogenies, you identify that multi-drug resistance is a common pattern across pathogens, and these strains are enriched in hospital environments. So I hope I have provided you some information about how we can do hybrid metagenomic assembly to get much higher quality genomes at strain resolution. We can use these to study resistance gene combinations and also disambiguate phages and plasmids with the host that they belong to. So all the work that we do is usually very freely available. All the data is freely available. The software, the method that we call Opera MS is freely available. So if you wish to give it a try, please do so, and we would be happy to help you do this sort of analysis. Okay, so I want to acknowledge my collaborators at Tan Tock Seng and NCID, Dr. Banimi Young, Dr. Kaleswar Marimutu, Dr. Untek, Brenda, Tim, and David, as well as Denis Bertrand, who was instrumental in this project. He led this project, and collaborators at SRIS, as well as Oxford Nanopore. So with that, thank you for your attention, and we're happy to answer questions.
Hi. Thanks, Dr. Nagarajan. I have a few questions, actually. First one, I think, I'm not too sure whether you may have any answers to this, but in your study of finding the CRE in household context and all that, do you find any association with food consumption and any particular sort of food that particular household may be eating?
That's a very interesting question. So in this, the study that I described here, we didn't have, the number of subjects was relatively limited because we're doing metagenomic studies, and the food data therefore was sparse relatively. But I think once you have, and also then we didn't have the food samples, we didn't have any microbiome data from those food samples. I think this is something we would really like to do in future studies, where we try to really relate the food and the environment of patients and the gut colonization. So some of the assemblies that I talked about, the high quality metagenomic assembly from the gut samples, can serve as references that we can map, say, analysis of food samples to, and try to relate that whether the strains that we find in food are really the same strains as what we find in the gut.
Thanks. I have a second question here from the audience. For environmental microbiome, is there a preferred extraction protocol, and would this skew results?
Okay, that's a good question. So for environmental microbiomes, there is a concern that you might miss, the profile might miss some of the hard-to-lyse bacteria. So we tend to use the same protocols as we use for gut metagenomic samples. So and that with bead beating ensures that we get an even coverage of DNA. But in terms of sequencing, I think the approaches are very similar for what we do with stool samples, and it's only when we are trying to do long read sequencing and the low biomass of the environmental samples that we face a challenge. But of course, there have been substantial advances in both PacBio and Nanopore sequencing in terms of working with low biomass samples. So the current protocols do allow for deep sequencing with even low biomass samples. So this is something we're very excited about, And I think in the future, we would not need to do enrichment cultures.
Thank you. I have one more question for myself. So I know I brought this up in the earlier talk, but you've done the hospital microbiome research. How do we translate and utilize these results in the hospital setting? I mean, we know that we use a lot of antibiotics. We know that subsequently there's going to be an increased number of drug-resistant organisms. There's going to be outbreaks of them. How do we utilize this hospital microbiome results that you have found?
Well, I think at the end of my talk yesterday, I emphasized the fact that the previous study did not have enough information about patient microbiomes, and also isolates from patients, infectious isolates, to really definitively establish a relationship between infections and patient colonization and the environment. But I think that's the first step. And then when we understand what fraction of what's out there in the environment and what specific subtypes infect patients and colonize patients, I think that's already a starting point, so that we focus more on being more concerned about those. But also having a sense of what are the environmental reservoirs for antibiotic resistance might tell us in terms of the antibiotic usage that we have. They might give us an early warning of what's getting enriched, but because presumably the patients are also seeding back the environment. Whether or not we see this directly in the patient isolates, we can see this in the environment certainly, that there is actually a reseeding process. So I think just like people have talked about doing this for sewage samples, using that as a basis for a surveillance of the whole hospital. But if you want to know ward by ward information, I think environmental samples from a particular ward might give you a sense of how much the treatment strategies that are being used might be shaping the environmental microbiome. I think finally, if people are interested in trying different environmental cleaning approaches, this is a readout, right? And you might combine that to say, are the cleaning measures effective? Are they eliminating the things that we want to eliminate? Or are they enriching them instead?
Okay, thanks. We have one more question. Were analyses for Candida performed for environmental microbiome studies in hospital? And if so, any specific problems with diploid genomes, and how did you overcome this?
Yeah, so in this study, we did not have that many Candida genomes. I think some of it might be the culturing approach that we used. In terms of assembly, yes, you're right. Diploid genomes present a bit more of a challenge, and now diploid genome within a metagenomic sample can be quite tricky. What would typically happen is that these genomes would, unless there's substantial divergence between the two copies, the two parental haplotypes, they will usually get collapsed into a consensus genome for the diploid organism. There are ways to tease those apart. So once you have the consensus genome, there are computational approaches to tease apart the two parental haplotypes. But that this is an area which has not seen much research and much work yet. If you're interested, we'd be happy to discuss that further.
Thank you. That's all the questions that we have. Thank you very much, Professor Niranjan.